coming and welcome to this week's seminar. I'm very pleased to introduce our speaker for tonight, Dr. David Denlinger, who is awesome to accept our invitation to speak to us this week. Dr. Denlinger is from Pennsylvania, grew up on a farm there. He got his PhD from the University of Illinois in entomology, after which he traveled around the world and did research in several different countries and Harvard University. He eventually ended up at Ohio State, and that's where he currently resides. His lab mainly focuses on insect overwintering and the mechanisms involved with that phenomenon. There is a social after this seminar at Ted Evans' residence. There are maps at the back of the room for those of you who don't know where that's located, so pick that up on your way out. Without further introduction, okay. Dr. Denlinger.
Thank you, Sam, and thank you for this opportunity to visit Utah State. I certainly enjoy the opportunities to meet with a number of you. I'm a little too loud. What do I do?
Yeah, maybe just turn it off and
turn it off.
Yeah.
I'm pretty loud anyway, so I think you'll be able to hear me.
Oh, do you have the pointer?
Yeah, I get that. Whoops, what'd I do?
Oh, fine.
And I can't, oh, there we go.
Okay, I think it's up.
What did
And, oh, sorry, we thought we got this figured out. Okay.
All right, I must have pushed the wrong button.
Oh, you're fine.
All right, okay, take two. All right, thanks so much for the chance to be here. I had a little chance this afternoon to sample the beautiful environment in which you live as well, and I can understand why you like to be here. It's a neat place. Well, as Sam indicated, much of my research in my home lab focuses on overwintering mechanisms for insects. And a few years ago, we were sitting around coffee and said, you know, if we're serious about this, we should try to push this to the extreme and see how an insect lives in a really extreme environment. And, you know, Antarctica might qualify for that. Now, we were aware that there were reports of an insect that is endemic to Antarctica, a little midge called Belgica Antarctica. It was discovered in the early 1900s in a Belgian research vessel called the Belgica was plying the waters of Antarctica, and a naturalist aboard found some of these midges, took them back to Belgium, and gave it the name Belgica Antarctica after that ship. Well, we were also aware that NSF has a polar program that supports specifically research in the polar areas, the Arctic and the Antarctic. So, frankly, I was a little nervous because I had never written a grant proposal for an insect I'd never seen before. And my biggest fear was they were going to fund us. We go down there and find the midge was no longer there or wouldn't be able to find it. But fortunately, that was not the case. We did get funded, which was good. And we went down and yes, we could find the midge where we were hoping it might be. So the next slide, just, you know, Antarctica, you know, it puts it in a little bit of a perspective here with North America. It's a little hard to really grasp how big Antarctica is when you just see it there at the bottom of the globe. But this is, you know, gives you some idea. I mean, it's really quite a bit larger than the continental U.S. Now, most of the nations of the world actually have stations in Antarctica. The U.S. operates 3 bases there. Palmer Station, which is where I've been going on the Antarctic Peninsula, also South Pole and McMurdo. These are both fairly large stations, and a lot of geological sciences and atmospheric sciences occur there. And to get into these stations, you can fly there from, they have airfields, you can fly there from New Zealand. The Palmer Station is much smaller, and we have to go in by ship. But one of the neat things about Antarctica, I mean, it was the fact that, you know, many countries have stations there. But in the 1950s, the nations of the world signed the treaty, the Antarctic Treaty, which says that Antarctica belongs to no country, And the only thing you can do there is have research activity that is for the betterment of humankind. There can be no activities for economic gain. There can be no military activity there. So this is my kind of a continent. I really like this place. Now there have been countries that have tried to lay claim to it, but that's a story for another day. But these are the 3 U.S. bases, and in order to get to ours, we have to first fly down to Punta Arenas, Chile, and there we get outfitted with our, NSF has a warehouse there for our extreme weather gear. We get all that there, and all of the experiments that we need to plan our experiments well in advance. I mean, I'm used to, at home, we need something, okay, we ordered, we get it the next day. Well, when you're working down there, you can't do that, so we have to put in the orders for all the chemicals and all the supplies we need about 6 months in advance. So that calls for a little more planning than I'm used to doing, but it forces you to do it. And actually, the first year we went down, arrived in Punta Arenas and found out that the bottle of Trizol that we had ordered had expired. It was bad. So the Raytheon Corporation that runs the station actually flew a plane back to Santiago to get us a bottle of Trizol to come back. We couldn't have done our step 1 of our experiments if we didn't have good Trizol. So it was the stations are run actually very well. They make sure that you, at least if you alert them of what you want, it'll be there for you when you get there. So this is the Lawrence Gould, the NSF's icebreaker that takes us down to Palmer Station. This is our first year down. A couple of my postdocs, Joe Reinhardt, Scott Hayward. Scott's now professor in Birmingham, England. Joe's a USDA employee at Fargo. So here we are. You first take off through the Straits of Magellan, down along the tip of Tierra del Fuego, and this is where down. Okay, now we're along the Antarctic Peninsula. Now, what I'm not showing you here is between there and here is the pretty rough passage. The Drake Passage that really 20-foot waves are routine, 30 or 40-foot waves are not at all unusual, and you really wonder, why am I doing this? There's a period in there of about a day and a half or so when you really do have to question why you're going through there. You strap yourself into your bunk and suitcases are flying all over the place. The cook is not very busy for a couple of days, as you might imagine. In fact, coming home the last time, one of my postdocs was on an IV the whole time back from Palmer Station. So it can be a tough ride and sometimes better than others. So this painting by Carmichael Wilson sort of portrays a time when I was unable to get out and take pictures myself. But okay, this is where we're going. This is Palmer Station, just a small station. It's the smallest U.S. Antarctic base, just here in a little spit of land that's on Anvers Island. A little closer up, this is the main lab building. Very convenient arrangement. You sleep up here, you go down a flight of stairs, have your breakfast, go down another flight of stairs, and there's the lab. So an easy commute in every morning. This is a boathouse where we where the Zodiacs are tied up to and keep our number of supplies like that. So as I said, your Raytheon Corporation has a contract for running these stations, and they do a really good job of it. At Palmer, it's really a base that can only accommodate about 45 people, and of those, most of the people are Raytheon staff who are maintaining the staff, maintaining the facility, electricians, carpenters, cooks, and only leaves room for about 15 scientists there. So we've been going down with a group of 5. So during the time we're there, we're about a third of the station during that time. Of course, even before we're going, we have to get fairly, since it's remote, and the ship drops us off, and then we don't have any way out of there for another 6 weeks. We have to have a fairly extensive physical before we're allowed to go. And especially they look a little, once they get to be my age, they make us do the treadmill test and all that, and before they let us go, because if you were to have a heart attack a few days after the ship left, you'd have to wait another 6 weeks till they get you out of there. So they want you to be reasonably certain that you're in fairly good health. Okay, the lab, you're pretty well equipped inside. Actually, they had a really, I didn't expect the sort of facilities and equipment that they have in a relatively small space, but really, really nicely equipped laboratory. And again, if we tell them in advance what we want, it will be there. It wasn't very satisfactory for doing, say, a lot of detailed molecular studies, but we could even do some northern blots and some things like that. But pretty well geared toward doing a lot of the physiological experiments we wanted to do. And this is why we went, to study the largest terrestrial animal that lives year-round on the Antarctic Peninsula. Okay, I mean, there are things like penguins and elephant seals that are bigger, but they just come up on land for a couple months or so at a time. And but in terms of year-round residents, this is the biggie. This is the larva of Belgica Antarctica. This would be the fourth instar larva. And it spends much of its life encased in ice most of the year. But when it thaws out in late December, then it feeds on algae and bacteria that are in that area. And then it freezes over again in early February until the next year, thaws out again, feeds again for a couple, month and a half or so, freezes over, and then the next year it emerges as an adult. It pupates and has a pupal life of about a week, and then the adults come out. This is a female on the right, a male on the left, and they're wingless midges. As there's a lot of wind in these islands that they live on, and as you have in many situations where you have windy islands, the insects lose their wings. So it's a very common arrangement on windy islands. Certainly if you had wings that became airborne, you'd go off to sea and would never come out again. So the females in males do not feed at all. It's a non-feeding stage. They simply come out, mate, lay eggs, and then die within about a week. Midges around here form swarms or flying in the air. Well, without wings, you can't do that, but they form swarms over the rocks and crawl over the rocks. It looks like a swarm, but it's just land-based swarm that they form. So the midge itself is not. This is Palmer Station right here. The midge isn't there, but it's out in these offshore islands that we see around here. So we go out to these offshore islands to get them. This blue line indicates a boating limit. It's sort of about a 5 mile radius from Palmer Station. We're not supposed to exceed that when we go out. But we go out in these zodiacs. This is Nick Tietz, one of my former graduate students, who's now just joined the faculty of University of Kentucky. So we get trained in operating the zodiacs, and as I told the group at lunch, I mean, one of the big tips is that if you go overboard, hold your hands like this so they freeze in that position so they can pull you out. So nice little tip you might want to remember should you find yourselves in those waters. So I mean, that was enough to scare me try not to go overboard. So this is Nick Shaving. So we go out to these islands in these zodiacs, and often might be ice like this that we can easily make our way through in the zodiacs. But then it changes so quickly. Within a few hours, all that ice might be gone, and we're supposed to keep in radio contact about once an hour. We're supposed to call back to Palmer Station to report that we're still alive and still doing well, and likewise they keep us informed if there's some weather front coming through, because things can change really quickly. This is sort of the habitat where we'd go looking for the midges. In these, this is Joe Reinhardt, postdoc at the time. See this green algae down here? This is Prassiola crispa. And usually we can find these midges really close to the penguin rookeries. Just we think that that high nitrogen content that's off the penguin rookeries is important for fostering the bacterial colonies that these midges are feeding on. They're feeding on the bacteria. They also will feed on this algae. And there's a lot of nitrogen. This is an elephant seal here. The elephant seal wallows generate a lot of nitrogen too, so a lot of bacteria from that. So if we can find an area like this, that's where we will go to work. One of the neat things about Antarctica is that, much like the Galapagos, the animals are not at all afraid of you there, in spite of the brutalities that occurred to them when their elephant seals and penguins were being collected for their oil. I guess that was a brief enough time that they were not able to, they didn't develop a fear of humans at that time. So we're not supposed to go real close to the penguin rookeries or the elephant seal wallows, but they haven't read that message, and they come over and look at us, and they'll come over and check us out, and that's allowed. So, that happens. And I guess the other thing I should probably say here, these penguin rookeries, they look so pristine when you see these beautiful black and white birds, and in a National Geographic picture, they look so great. But I always thought they should come with a scratch and sniff in those pictures, because they really smell awful. It smells like a pigsty around them, and you have to wash all your clothes after you've been out, even working, even this close to them, because they really take on quite a smell. This slide shows, I have a slide here of several different islands showing diversity of the different islands. The people here, Rick Lee is a long-term collaborator from Miami University. Again, this is Joe and Scott, who were there in our first year down there together. But you see this particular island has quite a bit of moss beds on it. Again, the Prassiola crispa down here is true in that one. This is a picture of including Luke Sandro, who was a high school teacher. Every year, in our team of 5, we included a teacher with us. In the first year, Luke was a high school teacher and a biology teacher from Ohio. And in NSF proposals these days, you always have to check off that box of broader impact statements, and this is our broader impact statement. We take a teacher with us, and the teacher's responsibility is, the main thing is to try to communicate back with the schools in Ohio and across the country who are interested in following what we're doing. They've always developed really neat blogs and really have engaged the school kids back home. But we also train them as scientists too, and so they can participate. Luke, we trained him to run an osmometer so he could take osmotic pressure of readings. So we try to train them in some scientific area as well, so they can be part of our scientific team too. Okay, there's just some scenes of general area. A lot of good bergs down there. There's a calving event. This is just behind the station. When I first heard the loud claps, I thought, a thunderstorm, but no, that's not what it was. We're hearing these all the time as the glacier recedes. Another mountain behind the station. And this is another island. In this case, one that looks a little fairly different. This Nick Teets here, and just a very, very rocky island in this case. And this, when you're there, it looks so tempting to just take off across the glaciers, but we really can't go very far because there's risk of crevasses, and there have been people who, of course, have died down these crevasses, and so we're somewhat restricted to just going up a glacier that's just behind the station. Beyond that, it becomes a little too risky to be wandering on our own. But people do come to visit us down there. This is I believe, a French sailing ship that paid us a visit while we're there. And indeed, if you happen to be in the area, you're most welcome to stop by. You can stop in at the station. We'll give you brownies and a cookie, and you can even buy a T-shirt in the gift shop. So, hey, we're not above commercial exploits here. And the T-shirt sales and the gift shop sales support the entertainment budget for the station, so it's a good cause. But we've had a number of really interesting people stop by. I mean, I can't imagine what it's like to go through the Drake on a ship of that size. But when you hear of the old sailing vessels, I mean, Darwin was deadly sick from the time they went around the tip of South America until he got to the Galapagos, and I think I understand why. Okay, so, and then some of these bigger ships, like the Volendam and Rotterdam, also come down. Now, those smaller ships, people can, like the National Geographic Endeavour and those ships, people can get off those and do some exploring on land. But the people that come in, by the thousands on these huge ships, they cannot get off. But we're asked to go out to those ships. NSF likes us to go out and tell our story, what we're doing there, why we're there. So we, in fact, this is sort of the scariest thing I've ever done when I'm down there. You go out there in these, little Zodiac, and they throw a rope ladder over the side, and you try to catch that at the right time of the swell and go up the ladder. And meanwhile, these tourists are there taking your picture. What these crazy people do, where they come from, and what are they doing? But then we get up on stage and do our spiel of tell them what we're doing, and try to convince them that their tax dollars are being well spent by our being there. And of course for us, after you've been there for a little while and at the station, you soon deplete all the fresh fruits and vegetables. So we usually come back, we go out there and have these wonderful buffets that we gorge ourselves on after we give our talks, and usually go back laden to the lab with freshies and cheesecakes that are absolutely wonderful that we wouldn't get otherwise. So it's a good thing in whole. Okay, some of the other organisms beside Belgica that live down there. This is one of my favorites. This is a tick, Ixodes uriae, the seabird tick. You can see them here underneath the rocks. This is a species of tick that comes out when the penguins first come into their rookeries and take a blood meal and then feed for maybe about a week or so, and then crawl back under the rock for the next year. And come out again. But one of the really neat aspects of this tick is it has a bipolar distribution. It's up in the Arctic, it's down in the Antarctic, nowhere in between. And people who have looked at them closely, really cannot distinguish those two populations. We really don't know how they got there. There are, of course, and it had to be some of those bipolar birds that fly from the Arctic to the Antarctic, things like the skuas or maybe the Arctic tern that make those huge migrations. But still, it's a little puzzling because the ticks on the penguins just they stay on the bird just for less than a week. So I don't know, some aberrant ticks must have decided to cling on to some of those birds and make that really crazy trip at some point. There are also a number of little mites. This is a little black mite that we often find in big groups here, Lascosites antarcticus. And there are several different species of Colembula that are there too. So these are close relatives of insects, not truly insects, but very close relatives. So several of those. And some of these are egg masses. Sometimes we see them in really, really huge numbers as well. We've done a little bit of work with both the tick and the mites and actually the Colembula, but certainly most of our work is focused on Belgica. Okay, some of the other animals, the elephant seals that, indeed, sometimes this is our lab door right out here. It comes up to check us out periodically, and again, we sort of let them do that and give them their space. This is a leopard seal. He's the top predator down there, and we have to give it the space it needs. So this is one in the water, and you're out in one of these little air-filled Zodiacs, and this guy with his really big, sharp teeth comes up to you. We've had one come up and check us out on one side, then disappear, come up on the other side, and it's eyeing you up for a potential meal. So we try to give them as wide a berth as possible. Actually, a couple of years ago, there was a British graduate student who was killed by a leopard seal while she was diving, doing some invertebrate work. It has this smile is a little Yeah, not really a smile, but it does look like it would be a friendly creature. But we try to give them the respect they need in the space, and they especially come in right in late January or February when the young penguins are about ready to take off and enter the water for the first time. These leopard seals congregate in fairly good numbers to pick them off as they try to learn how to swim. The fur seals start to come in mid-February or so. Lots of humpbacks down there. We've often been out, going from the station out to one of the islands and been delayed by going out to watch whales instead. And yeah, sometimes they come up a little closer than you'd like. These are a couple of my graduate students up here in another zodiac. And I mean, again, the whales are curious about it. They come up and really eyeball you. And it's just, from Ohio, I don't see whales that often. So it's been fun to experience them so close up in many times. And this is a dominant penguin down there, the Adelie. Colony of them here. And yeah, I don't quite get this, but late in the season, the chicks become rather big. They're as big as the adults, but the adults sit on their heads like that. I don't know. I mean, obviously the chick can't see, but I don't quite get what that's all about. But that's the way they do it. And soon after that, the chicks will start changing color. And recently, chin straps are coming in too. They're actually a warmer weather penguin, but in recent years they've started to come into that Palmer Station area. And a Gentoo, also another warmer climate penguin, has started to make appearance there as well. And some sheath bills are there. And skuas, I don't know if you know these birds or not. I mean, this is a nasty bird. I always thought if I were Hitchcock and do a really authentic film on the birds, you really should have used skuas, because these guys do dive bomb you. You're out there and they will come and attack and right at you, and especially during the breeding season, and fly very fast, right down to you, and kind of scary actually. Giant petrels. There's been quite a bit of work done with many of these seabirds by Bill Fraser's group working out of Palmer Station. They worked there for years and years. And a bird like the giant petrel can live 60 years or so. So the first years, Fraser's group just they become close to the birds. They wouldn't really touch them, but after a few years, then they would get to the point where the birds would accept them, and they could easily go in, lift them up, and take their eggs, weigh them, and measure them. And likewise with their chicks. These are birds that when they're off the nest, or flying up and down the coast of Africa, but they come to these islands to nest, as do many of these other birds that we've looked at. There's the Antarctic tern, again, that comes to these islands to nest, and the blue-eyed cormorant that nests in that area as well, again with a couple of its chicks here. So those are the kinds of animals that we encountered there. Now I'm trying to tell you a little bit about some of the research that we've done down there. This is just the segue into some of the more molecular sorts of things and questions we've asked. I mean, we did have an ice machine there, but my postdocs thought it was cooler to chill their tubes on 10,000-year-old ice instead of ice from the ice machine down the hall. That was just a photo op. We didn't normally do that, but that was the basis of that picture. One of the first questions we asked is if these insects have the ability to rapid cold harden. A few years ago, we demonstrated a phenomenon we call rapid cold hardening that is demonstrated here. Like if our fly in the temperate region is held at 25 degrees, put it to minus 10, they die right away. But if you have them at 25 and put them to 0 for as short as 10 or 20 minutes or up to an hour or two, and then put them to minus 10, they do very well. Some very, very, very quick response that occurs at an intermediate temperature that provides protection to an even lower temperature. Well, we asked if something like that could be operating in these midges as well, and indeed it can. We're plotting here survival, at this case at At -10 for 24 hours, they do very well. -15 for 24 hours, yeah, it's starting, they don't do quite as well. But if you first put them at -5 for 1 hour and then put them to -15, you see they do very well. And they're already in a frozen state at that point. So here's some really rapid event that's occurring when the animal is actually frozen. At -20 for 24 hours, they don't survive at all. But -5 for 1 hour, they can handle -20. So there is some sort of a very rapid cold hardening response that's operating in these insects as well. This slide just shows some different tissues of the insect. In this case, the dyes that we use, green means the tissue is healthy, red means it's dead. So here, the fat body, sort of the liver of the insect, if you will, at 4 degrees, which is pretty normal winter temperatures down there, they do very well. -20 for 24 hours, you see it killed it. -5 for 1 hour, and then to -20, you see most of the tissue was happy. So the fat body seems particularly vulnerable. Some of the other tissues not quite as vulnerable as the fat body and the gut are to this chilling response. I don't want to go into this in too much detail, just here I'm making the point that calcium seems to be very important for this rapid cold hardening response. In this case, we're looking again at cell viability, and these ones that have higher bars are ones where calcium was present. But if we use some pharmacological agents that deplete the calcium, we can see that in these frozen or rapid cold hardened animals, without calcium, they don't survive very well. So calcium appears to be a really important component that's involved in this rapid cold hardening response. Well, this slide of just showing ice crystals is here to remind me to say, well, the real challenge, the real problem with ice formation in the cell is that it ruptures the cell. You get the physical disruption that disturbs the cell as the mechanical formation of ice occurs. So you have to deal with this somehow. And one nifty way around it that these insects use is to simply get rid of that water. Most insects can maybe lose 20 or 30% of their water and still survive. These guys can lose 70% of their water and they still survive. They look like dried up little raisins. You can't imagine they're still alive. You pour water on them, they plump up and go on their merry way. So they have this remarkable ability to dehydrate and still survive. And what we see here, this is survival at -10. You see that if they're fully hydrated, they die, 48 hours, man, they're not making it. But if, in this case, just lost 30% of the water, they're perfectly happy. So once you've gotten rid of some of your water, in this case only 30%, you can go down the whole way to 70%. Once you get rid of your water, then you can survive at the lower temperatures. So this is one of the really key features that they figured out a way to live in a dehydrated state very nicely. There are some of the things we thought might be important, such as antifreeze proteins, no evidence for them. Many of our temperate region species will elevate trehalose, one of the blood sugars, or a polyol like glycerol. Well, there is a modest elevation of both sugars and glycerol, but this is very modest by comparison with what many of our temperate region species do. So, it's probably making a small contribution, but it's not the big player. Also, they'll clump up a bit like that when they, in response to desiccation, when they're in a cold hardy state too. So these are happening. Well, another group that we thought might be interesting candidates are the heat shock proteins. Now, these are proteins that actually we had worked on, commonly called stress proteins. We had worked on them in our temperate region species that overwinter, and we had shown that many temperate region species, when they're in their overwintering diapause, will turn on these heat shock proteins. And we've done experiments where you can knock down those heat shock proteins, and when you do that, you lose cold tolerance. So cold tolerance is really important in temperate region species during the winter. So we were interested to see if that might be the case for these insects as well. There are a number of different heat shock proteins. They're have classified according to their molecular weight. HSP70, a really dominant one. There's also a cognate 70 group. then an HSP90, and then a whole bunch of these small heat shock proteins that we've fished out of Belgica and are present in all kinds of insects. So these heat shock proteins play important roles both, as a protein might be exposed to heat or cold, it can denature, and then you take it a little bit further, you get a protein aggregate forming. And these heat shock proteins can act at both of these stages, both to prevent and repair protein damage that might occur. And we think, in the Antarctic situation, that's really important to keep maybe enzymes active and functional. And then if it goes a little bit further, you do have denaturation occurring. They target certain proteins, certain aggregates to get rid of them. So what you normally see is that these heat shock proteins come on in response to a high temperature stress or other kind of stress, and indeed, but they're not on all the time. And that's what we see here in the adults. I'm comparing here 4 degrees, 30, and 35 for an hour. And the adults show a very typical heat shock response. They're not on at the normal temperature, but they come on in response to a stress temperature, and then you go exceed that, and then you start killing the animal and you lose the response. So the adults, for both the small heat shock protein, HSP70 and HSP90, have that response where they're not normally on. But the larvae, even at 4 degrees, small heat shock protein is on, HSP70 is on, HSP90 during the normal conditions, and they continue a number of different conditions. Once you get up here to some really high temperatures, you start killing the animal, and they don't do very well. But so here's a case where these heat shock proteins are being turned on all the time, and we think that it probably plays a really important role in their cold tolerance, by analogy with our RNAi experiments where we can't use RNAi in these guys, but for our other temperate region species, when we knock them out, we lose our cold tolerance. So we think that's an important adaptation for these guys as well. Another challenge that they face in Antarctica is the really high ultraviolet levels, the ozone hole, but even without the ozone hole, there's still a lot of ultraviolet radiation occurring here. And so we've looked at some of the ROS detoxifying enzymes, like catalase and superoxide dismutase. And again, the short story here is, like the heat shock proteins, these enzymes, the genes encoding these enzymes, are turned on at pretty high levels all the time. Adding more sunlight doesn't increase, so they're already being made constitutively all the time. So these guys can tolerate a wide range of stresses. You can stick them in seawater for 6 days and they survive very nicely. Most insects, you stick them in there for 15 minutes or so, they'd be dead. This is a natural challenge that they have to be faced with, because these islands are often in locations not too far from the shore, and big storms like this come up and bring seawater up over the land. And of course, then when they're frozen in ice, fresh water, they have to be able to tolerate ice melt of no osmotic pressure, and as well as seawater. So they can handle a lot of different stresses. Well, we decided that for a lot of the work we wanted to do, we wanted to go after the genome of this so we could really have access to the genes that we wanted to work with. And the real surprise to us when we sequenced the genome of Belgica, and it had an assembled genome size of 89.5 megabases, which was the smallest genome reported. And this is the first genome we did, so we figured we don't know what we're doing. We just missed a third of it or half of it or something like that. But we went on to look at flow cytometry, and indeed verified that the estimated size is about 98 megabases, making it, these are different estimated genome sizes of a whole bunch of insects. So Belgica is way down here at the smallest genome size. Median dipter size is about 235. Previously, the smallest genome was that of a body louse, but it's, again, considerably larger than Belgica. So how do you get this small size, and what's this happening? Is it because it's a chironomid, or is it because it's an extremophile? There really had not been any other midges or chironomids that had been sequenced before this one. So, it could be, well, maybe all chironomids are really small like this. But we have now done some flow cytometry of a number of other chironomids, and they are all kind of small, but not as small as Belgica. So I think it's both a function of it being a chironomid, which generally have small genomes, but probably also of it being an extremophile. So how do you do that? I mean, they consistently, very small introns, very few repetitive elements, and those few transposable elements that are present seem to be old and not active. There's sort of enrichment of things involved in membrane-related things, and ATP binding, and things like odorant receptors and that kind of thing are not very well represented in that genome. But it has basically the same, about 14,000 functional genes. This is what most insects have. It's just gotten rid of all that other junk that most insects and other organisms have in their genomes. Okay, with the genome, we of course were able to do some transcript work and really ask some of the questions we wanted to know of, what are the big things that are happening, especially in relation to desiccation, which seemed to be such an important trait. So we've actually done a couple of different kinds of desiccation, and I'm not going to, one we call cryoprotective dehydration. It's a different way of desiccating the animal in the exposure to ice, and the other is simply putting into a 93% relative humidity for 4 days. In both cases, you get about a 40% water loss. But the big story here, the story here was there really wasn't much difference between these two. So I'll just show results for this form of desiccation. And then we measured gene expression using RNA-seq to see what kinds of changes you would find. And just, again, to capsulize this in a little way, you see some very, very dramatic changes that are occurring. Things like this autophagy pathway, the green means highly upregulated. So there's a lot of really cool things happening here that are emphasizing autophagy, which means things that are involved in recycling processes of the cell. Whereas apoptosis, cell death, those mechanisms are shut down. So you're conserving many of your organelles and the tissues within the cell by these autophagy pathways during desiccation. And then you also have this, the red again means it's been shut down. So major metabolic events like glycolysis and the TCA cycle and genes involved in ATP synthase are very highly shut down. So you have this dramatic metabolic shutdown that's going along with the desiccation process. We've also done metabolomics experiments which nicely complement the transcript studies, all showing a very consistent story of these kinds of pathways being, autophagy being up and metabolic processes like this being shut down in response to desiccation. Well, we were quite interested in genes like the aquaporins. These are the pore canals for which water moves across the cell. We thought if desiccation is important, maybe these aquaporins could be really critical for the process of desiccation. Well, we tried previously very hard to get these aquaporins, and all we could ever do was come up with one of them, and we tried to clone them. But now having the genome, we had already reported this one, but now we have 5 additional aquaporins. And this is sort of the beauty of having access to a genome. You now have all the genes you might want to work with, and we're now trying to make some sense out of these aquaporins. Are they contributing? This is an aquaporin called DRIP that comes on pretty strongly in response to some dehydration responses here. This is really the only one we've characterized functionally so far, and that's one of the ones we're going to be looking at again real closely when we go down again. But one of the other questions, sort of this final set of experiments I want to talk about is, how does an organism respond to the seasonal or the daily cycle across the seasons in Antarctica? We're going there in early summer when it's essentially daylight around the clock, and of course it goes from that to almost complete darkness at the other time of the year. And how does the insect clock work and recognize those kinds of subtle changes that are, well, subtle within a day that are occurring? And is this important? So let me just go through a series here of different times. So this is January 8 at 8 p.m. at a location behind the station. This is 10 p.m. This is 11 p.m. This is sunset at midnight. And by sunset, I mean the sun just goes below the horizon at that point. You can still read a magazine or anything you want to under these conditions. It never gets dark enough that you can't do that. So this is sunset. This is 1 a.m. This is 2 a.m., 3 a.m., sunrise. So here we go again. Okay, 4 a.m., 5 a.m., 6 a.m., 8 a.m., 10 a.m., 12 noon, 2 p.m., 4 p.m., 6 p.m. A little rain came at that point, and so there we are. So you can see really in that particular day, it was really pretty hard to tell the difference between day and night. It wasn't so obvious. So with that in mind, we thought we'd like to see what the clock genes do in that sort of environment. And again, I'm not going to go over any details of this, but these are sort of the major clock genes that operate. Many of you have probably heard of the period gene, the timeless gene. They form a heterodimer that go into the cell and are part of the time-keeping process. And light is recognized by cryptochrome 1, and it plays an important role. So again, I'm not going to go into the details, but these are the major players, and we've cloned all of these genes, been able to get access to all these genes. And now the question is, how are these changing during the day? Now, in the temperate region, what you would see for a gene like period or clock, this is under 12 hours of light, 12 hours of darkness. Period and timeless especially peak right here at the beginning of the SCOTA phase and then drop. You can see under these conditions, they did not. They were just sort of, the gene was being expressed. It was there, the genes were there. They're being expressed all the time, but no cycling occurring at all, even under, in this case, somewhat unnatural photoregime, but no evidence for cycling. And we went into the field. We took little cameras and monitored locomotory activity of these midges, in this case under 3 hours of light, 3 hours of light, 3 hours of darkness, 21 hours of light at 5 degrees. And this is a monitoring locomotory activity of the larvae of Belgica. And you can see they're constant, even during the dark period, they're as active here as they are then. And next day, then we switch them complete darkness at this point. Again, they just keep going. They just keep going until it died. And when you look at the gene, the period gene, the timeless gene under those environments, just absolutely no change at all. So they keep on, the clock is present, but it's not showing this kinds of cycle that we anticipated that we would see in a temperate region situation. Well, at this point, the ship comes back to get us, pick us up again, and we can indeed take the midges with us. We routinely collect lots of these midges, bring them back to our own lab, and extend our experimental season. We can keep them very happy in our cold room at 4 degrees. If we bring them out to the lab at 25 degrees, they're dead in a couple of hours, but as long as we keep them at 4 degrees, and we've been able to keep them live and very happy up, we bring them back 1 February, we can keep using them through to the next December or so and extend our experimental period quite a bit. We haven't succeeded in actually rearing them in the lab. We get occasional adults emerging, but we haven't been able to propagate them. But then, we really haven't tried terribly hard. If we were to succeed, then we have no excuse to go back the next year. But this next time, I think we are going to try, put a little more effort into trying to see if we can rear them. My Japanese colleague, Shin Goto, a former postdoc who's been with us, has had a little bit of success rearing them on some algae that he can get in Japanese grocery stores, and they seem to like that pretty well too. So, okay, what do we just talk about here? How do you survive in Antarctica? And these are the lessons I'd say we've learned so far from Belgica. Okay, get rid of your body water. Hey, then when you do that, you can handle the low temperatures. Turn on your heat shock proteins, turn on your antioxidant responses. Okay, elevate your trailers and glycerol and even proline a little bit. That probably helps a bit. And you boost those recycling pathways, the autophagy pathways, shut down your metabolism pretty dramatically. Then simply be an opportunist. Who needs a clock? I mean, you have such a short time to live. You just go flat out the whole time until you die or freeze over again the next time. So there's really such a small season that they can actually exploit. So I think they simply adopted that mechanism of going full out the whole time, night or day. And I think the final slide just point out a couple of people. I don't know everyone who's been involved in this project, but current people. Drew Spock will be going down with me this year for the first time, a graduate student. Justin Payton was involved in some of the genome work of Belgica. Postdoc Elena Kovalkova did a lot of the clock work on the midge. Nick Tietz went down a couple of times, did a lot of the desiccation transcriptomic stuff, and also involved in the sequencing. Let's see, Rick Lee, longtime collaborator. He's been down every year with us. Shin Goto, a former postdoc of mine now at Osaka, is doing a lot of the work on the Aquaporins. Joanna Kelly was involved in the genome sequencing story too. This particular project was supported by the NSF Polar Program. And with that, I will close and thank you kindly for your attention. So if you please.